DNA replication in prokaryotes. DNA replication has been extremely well studied in prokaryotes primarily because of the small size of the genome and the mutants that are available. E. coli has 4.6 million base pairs in a single circular chromosome, and all of it gets replicated in approximately 42 minutes. Starting from a single origin of replication and proceeding around the circle in both directions. This means that approximately 1,000 nucleotides are added per second. The process is quite rapid and occurs without many mistakes. DNA replication employs a large number of proteins and enzymes, each of which plays a critical role during the process. One of the key players is the enzyme DNA polymerase, also known as DNA pol, which adds nucleotides one by one to the growing DNA chain that are complementary to the template strand. The addition of nucleotides requires energy. This energy is obtained from the nucleotides that have 3 phosphates attached to them, similar to ATP, which has 3 phosphate groups attached. When the bond between the phosphates is broken, the energy released is used to form the phosphodiester bond between the incoming nucleotide and the growing chain. In prokaryotes, 3 main types of polymerases are known: DNA pol I, DNA pol II, and DNA pol III. It is now known that DNA pol III is the enzyme required for DNA synthesis. DNA pol I and DNA pol II are primarily required for repair. How does the replication machinery know where to begin? It turns out that there are specific nucleotide sequences called origins of replication where replication begins. In E. coli, which has a single origin of replication on its 1 chromosome, as do most prokaryotes, it is approximately 245 base pairs long and is rich in its sequences. The origin of replication is recognized by certain proteins that bind to this site. An enzyme called Helicase unwinds the DNA by breaking the hydrogen bonds between the nitrogenous base pairs. ATP hydrolysis is required for this process. As the DNA opens up, Y-shaped structures called replication forks are formed. 2 replication forks are formed at the origin of replication, and these get extended bi-directionally as replication proceeds. Single-strand binding proteins coat the single strands of DNA near the replication fork to prevent the single-stranded DNA from winding back into a double helix. DNA polymerase is able to add nucleotides only in the 5' to 3' direction. A new DNA strand can be only extended in this direction. It also requires a free 3'O group to which it can add nucleotides by forming a phosphodiester bond between the 3'O end and the 5' phosphate of the next nucleotide. This essentially means that it cannot add nucleotides if a free 3'O group is not available. Then how does it add the first nucleotide? The problem is solved with the help of a primer that provides the free 3'O end. Another enzyme, RNA primase, synthesizes an RNA primer that is about 5 to 10 nucleotides long and complementary to the DNA. Because this sequence primes the DNA synthesis, it is appropriately called the primer. DNA polymerase can now extend this RNA primer, adding nucleotides one by one that are complementary to the template strand. Figure 1. The replication fork moves at the rate of 1,000 nucleotides per second. DNA polymerase can only extend in the 5' to 3' direction, which poses a slight problem at the replication fork. As we know, the DNA double helix is anti-parallel. That is, one strand is in the 5' to 3' direction and the other is oriented in the 3' to 5' direction. One strand, which is complementary to the 3' to 5' parental DNA strand, is synthesized continuously towards the replication fork because the polymerase can add nucleotides in this direction. This continuously synthesized strand is known as the leading strand. The other strand, complementary to the 5' to 3' parental DNA, is extended away from the replication fork in small fragments known as Okazaki fragments, each requiring a primer to start the synthesis. Okazaki fragments are named after the Japanese scientist who first discovered them. The strand with the Okazaki fragments is known as the lagging strand. The leading strand can be extended by one primer alone, whereas the lagging strand needs a new primer for each of the short Okazaki fragments. The overall direction of the lagging strand will be 3' to 5', and that of the leading strand 5' to 3'. A protein called the sliding clamp holds the DNA polymerase in place as it continues to add nucleotides. The sliding clamp is a ring-shaped protein that binds to the DNA and holds the polymerase in place. Topoisomerase prevents the overwinding of the DNA double helix ahead of the replication fork as the DNA is opening up. It does so by causing temporary nicks in the DNA helix and then resealing it. As synthesis proceeds, the RNA primers are replaced by DNA. The primers are removed by the exonuclease activity of DNA pol I, and the gaps are filled in by deoxyribonucleotides. The nicks that remain between the newly synthesized DNA that replaced the RNA primer and the previously synthesized DNA are sealed by the enzyme DNA ligase that catalyzes the formation of phosphodiester linkage between the 3' end of one nucleotide and the 5' phosphate end of the other fragment. Once the chromosome has been completely replicated, the two DNA copies move into two different cells during cell division. The process of DNA replication can be summarized as follows. DNA unwinds at the origin of replication. Helicase opens up the DNA-forming replication forks. These are extended bi-directionally. Single-strand binding proteins coat the DNA around the replication fork to prevent rewinding of the DNA. Topoisomerase binds at the region ahead of the replication fork to prevent supercoiling. Primase synthesizes RNA primers complementary to the DNA strand. DNA polymerase starts adding nucleotides to the 3' end of the primer. Elongation of both the lagging and the leading strand continues. RNA primers are removed by exonuclease activity. Gaps are filled by DNA pol by adding dNTPs. The gap between the two DNA fragments is sealed by DNA ligase, which helps in the formation of phosphodiester bonds. Summarizes the enzymes involved in prokaryotic DNA replication and the functions of each. Section summary. Replication in prokaryotes starts from a sequence found on the chromosome called the origin of replication, the point at which the DNA opens up. Helicase opens up the DNA double helix, resulting in the formation of the replication fork. Single-strand binding proteins bind to the single-stranded DNA near the replication fork to keep the fork open. Primase synthesizes an RNA primer to initiate synthesis by DNA polymerase, which can add nucleotides only in the 5' to 3' direction. One strand is synthesized continuously in the direction of the replication fork. This is called the leading strand. The other strand is synthesized in a direction away from the replication fork, in short stretches of DNA known as Okazaki fragments. This strand is known as the lagging strand. Once replication is completed, the RNA primers are replaced by DNA nucleotides and the DNA is sealed with DNA ligase, which creates phosphodiester bonds between the 3' of one end and the 5' phosphate of the other strand. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.